Hi,
Great.
everybody. I'd love for you all to put where you're from in the chat. Just to remind you that if you have questions for our presenters, please be sure to put them in the Q&A function of your Zoom. But if you want to talk to one another, you want to shout out to me, great. Go ahead and put that in the meeting chat, and we can have fun there. But again, questions should go in the Q&A. That's what we'll be monitoring before we get started. And we are at the top of the hour. So if you guys are all ready, I think we can go ahead and get started. We are recording this session. So here we go. Good afternoon, good morning. To those of you where it's morning, to everyone, and behalf of the FSHD Society, welcome to today's FSHD University webinar. Again, before we get started, just a quick reminder, this session will be recorded. So I had some questions in the chat already about how they can access this webinar. It will be recorded, it is being recorded, and it will be made available on our YouTube channel within a week or so. And again, encourage you to use the chat to interact with one another today, but please use the Q&A function for any questions that you'd like to ask our presenters. Today, we are so excited to bring together leading experts to explore how gene therapy is being developed to target the root cause of FSHD, including how it works, why it holds promise, what patients should consider when thinking about clinical trial participation, such as eligibility, safety, long-term follow-up, all of those sorts of things. We'll also discuss the role of adeno-associated virus, or AAV, as you've probably heard it referred to. That's the delivery method used in gene therapy. It is a delivery method, not it's an A delivery method. We're pleased to welcome our colleagues from Epicrispr today, who will share updates and answer your questions about Epi 321, which is their investigational gene therapy for FSHD currently in development. But now it's my personal pleasure to introduce today's presenters. We have Dr. Lucienne Ronco, is our Chief Science Officer here at the FSHD Society. She leads strategy across research, innovation, global collaboration, all for FSHD. Dr. Lawrence or Larry Hayward is a professor at UMass Chan Medical School and is internationally recognized for his decades of research on the genetic and molecular mechanisms of FSHD. And Dr. Wes Miller, the Chief Science Officer at Epicrispr, who is leading the development of next generation epigenetic therapies, including Epi 321 for FSHD. So we are so grateful to all of you for being here today. And I would like to just turn it over to Dr. Ronco.
Thank you, Beth. Thanks for the great introductions and putting us in a good place for understanding what we're doing today. I'll say a couple of things also as introduction, and then I'll speak about, at a high level, about these adeno-associated viruses. And from here on in, we will call them AAV. It just makes it easier. And I'll also tell you, a lot of people have joined since we even began the discussion now, that I and Larry and Wes, we will repeat a lot of the information that we described to you today, and we'll repeat at different levels of detail. And I think that repetition is really valuable when people are striving to understand the complex world of making new therapeutics and of using AAV in the process. As Beth said, I'll reiterate, you can put your questions in the Q&A. We hope to address all of the questions that we pulled together ahead of time in our presentations, but also I'll go back and ensure that we've addressed most of the key questions as the moderator. And then you'll be able to view this again, as Beth said, it'll be on our website. So let's talk about the development of new therapeutics for FSHD. You know, we know many companies are developing new treatments to reduce or block DUX4 in muscle cells. It includes Avidity, Dyne, Sarepta, and there's a very long list. And we're seeing success with these approaches in the clinic, and it's making we drug developers and we patient representatives very optimistic. There are other groups that are working to stimulate muscle growth, either in the presence of DUX4, we haven't yet knocked it down, or in the absence of DUX4, where we have knocked it down with a different therapy. But we're not going to talk about those approaches today. We're going to talk about that at a future university. We just discussed this yesterday, the concept of regeneration and driving new muscle growth. We're not going to discuss that today. We're talking about targeting DUX4 and reducing DUX4. So it's a very rapidly moving field of AAV and viral gene delivery. And we're going to speak in general and then more specifically about FSHD. These viruses are tools to deliver specific therapies to specific tissues. So imagine a FedEx truck pulls up to your house, they bring the package up and they deliver it to your front door. These viruses pull up to a cell. In some cases, some of those viruses pull up to a muscle cell and they deliver their contents into the cell. And so they are delivery tools. There are types of AAV that go directly to muscle, and Larry's going to speak in more detail about this. And Epicrispr is using a version of AAV that does target skeletal muscle. While they're designed to target muscle, they do circulate to other organs, and we'll talk a bit about that. So the field is currently undergoing really significant advances in how we use viral therapies. Progress is happening in industry, in academia. And these viruses are constantly being changed in laboratories to try to identify new viruses that will enhance delivery and reduce immune responses. We'll talk about that. So in general, the field has seen successes and challenges. Some of these challenges pose unique risks, and they have unique benefit profiles also in muscle dystrophy. In the benefit column, we have seen safety and effective delivery based on AAV gene therapy. The ongoing risks that the field is continuously working to build more effective approaches are how do we adequately and deliver a highly effective gene therapy that targets muscle well? That's our big question for FSHD. How do we get a lot of virus in viral particles to deliver to a muscle well? And we then have to engage and understand how do we deal with the immune system and immune activation that's driven by the virus? Are there long-term consequences of AAV gene therapy? Honestly, we probably don't know yet. It's such a new field. Those long-term consequences are something we have to pay attention to. How durable or how long does the response last? And we don't have a strong answer for that yet. That is work that will be followed as the field matures. And how many FSHD patients have had a prior exposure to AAV? As we age, as human beings, we interact with other people. We have an opportunity to be infected with AAV that allows our immune system to make antibodies. And if we're in that situation, then we may not be. Beth, did you have a question?
Lucienne, are you intending to share some slides with us?
No, I'm not.
Oh, okay. I'm sorry. Didn't mean to interrupt. I thought we were looking at slides, but we're not.
No, Larry and Wes will have slides. I was just giving a high-level introduction. Okay. So have you been exposed to AV in the past? And in that case, you may not be able to participate in a study. So today I'm speaking in general, and Larry may speak in general, and then Wes will speak specifically about Epi321. The proposed mechanism is to block DUX4 from being turned on in skeletal muscle, and it does this by changing the DNA surrounding DUX4 and blocking the ability for cells to make it. In preclinical studies, high doses of Epi321 was found to reduce DUX4 expression and protect muscle tissue from damage, and it's delivered through this AAV that targets muscle cells. Wes will speak more about that. Epi has cleared an IND in the United States and a CTA in other countries, and they have been able to start dosing patients in the US and abroad. And Wes will speak more about that. So what's been seen in the clinic? Many companies are developing AAV therapies: Takeda, Pfizer, Novartis, AbbVie, Epicrispr, Sarepta. For AAV therapies in muscle dystrophy, there has been modest or moderate success. DMD has seen the most activity, Duchenne's muscular dystrophy. In 2024, Elevidys is the name of the compound from Sarepta, received full approval, despite, I have to sadly say, I think, unclear efficacy. There are different challenges in DMD than there are in FSHD. For DMD, they want to restore a protein called dystrophin, and they have to restore a good portion of that protein, and it's a big protein, and it may be hard to do. So it's been unclear how well they've restored a functional protein. They can see the protein, but is it doing its job? We don't know the answer to that. We also don't know how well the virus gets to the muscle and how long the virus can maintain its effect. Pfizer had a DMD approach, and they discontinued it because of efficacy and safety reasons. So some companies move forward. They've got some data that suggests move forward. Some companies stop because they see that it's not meeting their endpoints. The same thing will happen for FSHD. And so we have to be prepared for some companies moving forward and some companies not. More profound clinical success has been seen in spinal muscular atrophy, SMA. Multiple AAV therapies have been approved by the FDA. And there's a lot of work going on with AAV. There are seven therapies, seven AAV-based therapies have been approved in the United States. More than tenfold are in clinical development. So there's a lot of work in this area. A lot of patients have been treated in SMA, more than 4,000, in DMD, more than 600. So there's been a lot of work with this. And every time we dose another patient, we learn. And those learnings are getting wrapped into our new work with those molecules. A lot of clinical experience and a lot of research experience is changing and is driving our therapeutic success. The other thing that has to evolve is our manufacturing processes. So they're getting better. They should, as they mature, we should be able to reduce the dose required for efficacy, and reducing the dose is often associated with reducing toxicity. So for instance, Novartis, that has brought forward a very successful SMA therapy, they continue to modify their production of their dose so they can decrease the volume of the therapy that's delivered and increase the efficacy with that smaller dose. So you can anticipate in FSHD the exact same thing will happen. We'll start with a process, and that process will get better and better. Our challenge is that we've got to get high levels of muscle targeting for FSHD while we minimize liver toxicity and immune responses. I'm sorry to say that lethal toxicity in AAV gene therapy has been seen. It's rare, but it exists. It appears to be associated with high dose intravenous injection of therapeutic products. So we've seen vector-induced liver failure, kidney failure, heart failure, and recently lung failure. Immune responses to the virus have been seen, and cardiac involvement attributed to the immune response has been seen. I'll talk a little bit about hepatic toxicity. It's been seen in 20 to 90% of patients, and the severity is proportional to the dose of the drug. More severe outcomes seen in high doses and in pediatric patients. I would say, let me talk a little bit about Sarepta. They've been in the news lately. There have been rising adverse events and deaths have been seen. So they have a new warning, and they're doing a new warning, like a black box warning, death based on acute liver failure in non-ambulatory patients. How Sarepta is responding to this is they're planning an enhanced immunosuppression regimen. They must suppress the immune system before treatment and then after treatment for at least 2 months post-treatment. They may be being kept in the clinic for at least 2 months post-treatment. And there's also enhanced weekly monitoring. So this is what's happening in Duchenne's muscular dystrophy at this time. So gene therapy is an efficient platform for long-term transgene expression, and I'm sure we'll be talking about that with the possibility of having a single treatment and efficacy or that persists for a long period of time. So this is important work and exciting work for the field, and we are following it closely, and Larry is following it closely. And so now I'm going to hand it to Dr. Hayward and ask him to talk about his experiences and how he advises his patients. Larry.
Okay, thank you very much. I'm going to try to share my screen here. 1 second. Okay. There. Hopefully you can see my screen.
Yes.
Yes. Okay,
Yep.
great. Yeah. So, yeah, thank you so much, Lucienne. I'm just really happy to present to this group because I feel very fortunate to work with FSHD patients who overall are so willing to help in our research aspects and I think that has made a huge impact on accelerating the potential treatments that are coming down the line. Um, Let me see if I can advance here. Yeah. So this is just briefly to some of my disclosures. So I've worked on with three different companies on FSHD trials, and I've also just of note I've been a principal investigator for a different trial for a different type of CNS and PNS a neuropathy called adrenomyeloneuropathy that it was also an AAV study. Um, but what I want to just give a really sort of high level overview are the different levels at which one can envision a treatment for FSHD. So starting sort of at the DNA level, this is where the DUX4 gene resides and where what happens in FSHD is that because of decreased methylation, there's a tendency for this powerful DUX4 gene to come on inappropriately in muscle. And when that happens, it's toxic. And it can harm one muscle cell at a time, leading to effects on individual muscles over time that we don't really have a whole lot of control over. And we're still learning about what triggers it to come on in one cell versus another. But there are already there are thoughts about, well, can we intervene at the DNA level in the genome itself to try to correct the situation? So gene editing would come to mind for this, and this involves this CRISPR-Cas technology for the most part. And I give a couple examples here. One would be to correct a gene that's defective and preventing proper methylation. Another would be to actually go in and sort of do some microsurgery on the DUX4 locus so that you can't generate a stable DUX4 messenger RNA. And then another would be to somehow restore DNA methylation, and Wes will be talking more about that. If so, let's say you can't attack it at the DNA level, then what happens is over time, DNA leads to, when it becomes transcribed, it leads to the production of what we call a messenger RNA or mRNA. And this is simply, these are RNA nucleotides in a long string that would encode the instructions for making a protein. And what we're concerned about is making the DUX4 protein. But so there's been earlier research showing that there may be ways to sort of make it less likely that even if that gene region's open and you start making some transcript, that maybe you can make less transcript or inhibit it. And that was the basis for one of the Fulcrum trials looking at a protein kinase inhibitor. So it wasn't directly addressing DUX4 very specifically, but it was at least a valid hypothesis to test, can we just tamp it down a bit by inhibiting this kinase? As we know, that really in the trial, didn't demonstrate a definitive effect. But if we look farther down, now let's say we can't stop it from making an mRNA. Are there ways to develop a treatment that can act as sort of a surveillance mechanism for these mRNAs and lead them to be destroyed before they have a chance to be made into protein? Which is the actual toxic agent. And so there are a number of other studies looking at what we call either small interfering RNA or microRNA inhibition to silence and sort of target for destruction these RNAs. Now, as you go beyond this level where there's actually DUX4 protein, then you have to think about, well, what's the protein doing downstream? And then the problem is there could be many things it's doing, and it's harder to envision a therapy that's really going to be effective if you have to inhibit 10 other things. So the farther upstream you can get, potentially you've got something that's getting really to the root cause of the disease. And so, what I just want to give another a real like high level framework overview is uh, some of the main types of clinical trials. Because when I see a patient in my FSHD clinic and they express an interest in being involved in a clinical therapeutic trial, then I have to get across to them. Well, I want to get as much information across as possible, and what are the choices, and because each choice has a different level of potential risk versus reward. And I should emphasize overall, though, for all clinical trials, there's not necessarily implied a direct benefit to the patient. I mean, it's just so amazing that patients are willing to be enrolled in clinical trials for all the work it takes, and the time it takes for them. But many of these are placebo controlled, where, for instance, half of the patients will be getting a placebo and half will be getting the active agent. And I won't even know what the patient's getting. So we can't necessarily say that when you participate in a trial that there'd be a direct benefit to you. But what's often the case is that after the period of a blinded part of a trial, then often companies will offer the agent to the patient in what's called an open label extension. And that, again, that's trial specific, but just to keep in mind that there's no guaranteed benefit for any patient. So if we look at these are the three main ways I try to explain to patients different trials. So that fulcrum losmapimod trial would be an example of a small molecule drug. You could enroll in the trial, and if you're in the middle of the trial and you get some sort of adverse effect, you can just say, I'm just not tolerating this. I want to stop. I don't want to take it anymore. And then, the next day, you don't have to take it. In general, for those types of trials, any side effects would tend to wear off over time fairly quickly. The interfering RNA-based treatments, these tend to be an IV infusion of a nucleic acid that's coupled to something that will help target it to muscle. And so these also you could stop if it's not agreeing with you. It may hang around for several weeks in the bloodstream, depending on the agent. And it's a treatment that, like the small molecule drugs, would be something that would have to be repeated over time. So most of a lot of the thought is that like you need to get this treatment as an IV infusion every 6 to 8 weeks if it's shown to work. So it's by no means a cure, but these are potential treatments that can help. Gene therapies are in sort of a different class in that because we were talking about AAV, it's because of the immune response, it's sort of a one and done treatment. You can get it, at least at this point in time, you can get it once, but it would be hard to envision getting a second treatment with it because you would have developed antibodies. There is research going on to overcome that, but it's still a ways off. And the other side of that is that this agent is designed to last in the body for a long time. It could be years. And so there could be acute side effects from the agent, or there could be other unforeseen long-term side effects. So it's sort of a different category of treatment than some of these others. But I just want to go through a little bit about AAV. So my understanding is that, first of all, the AAV virus in the wild, many of us have been exposed to it. Many of us have not developed sufficient antibodies to it to pose a problem. But it's a non-pathogenic virus. Like you could get exposed to AAV and you'd never know it. It doesn't really cause any symptoms. There is a virus called adenovirus, which is a very common cause of upper respiratory infections, especially this time of year. And an AAV, adeno-associated virus, was actually found as a contaminant when people were isolating adenovirus. So it really doesn't have anything to do with adenovirus itself. It's just a virus that was identified because it happened to be in samples that were being examined from people who were sick with adenovirus. And yet it was found, it really doesn't cause any symptoms or problems. And so, but what it is it's simply a way of packaging DNA, which is what we want to deliver for this type of therapy. So the DNA is inside here, and this shell that it's in is called the capsid. And so we have ways in the lab under very stringent criteria to put in any DNA that we want to deliver into this sequence. And it has certain other elements from the virus that are important for maintaining it in the capsid and producing lots of these. But these don't cause infection. The actual AAV DNA has been removed and replaced with what we want to deliver. So it was already non-pathogenic, and now it's just delivery. It's used as a delivery tool. And what's key, though, is the composition of the capsid, because there are certain cells that have receptors that will recognize a type of capsid. And as Lucienne mentioned, the capsids are being engineered to be more specific to muscle. Like the standard AAV, it might go indiscriminately into many cell types, including the liver, where we really don't want to deliver a whole lot because of potential toxicity. So there are capsids being designed so that it'll be preferentially being recognized by muscle cells. And so when it binds to this receptor on the surface of whatever cell is able to take up the AAV, the membrane sort of invaginates a little bit, and it forms a little bubble and gets taken inside the cell. And I'm just gonna before we get to going inside the cell, I just want to talk about the AAV that's floating around out here. If there are antibodies to it, one of the concerns is if there's an antibody here and it recognizes this, then it can trigger a pathway of activation known as complement. So there's certain proteins that see this interaction and they say, okay, we've got to ramp up some immune reactions to this. And unfortunately, what that can do acutely is it can lead to damage of blood vessel wall cells through a very complex set of interactions here that can lead to disruption of the membrane. And that can cause these early effects, because this is only going to occur in the likely first few days to weeks while it's floating around the bloodstream. It's not going to be in the bloodstream for very long. And it can be recognized inside the cells too. But the major risk here is that you'd have complement activation acutely, and that you could have some damage to the blood vessels, and it can either lead to decreased platelets or changes in blood clotting or even effects on the heart. So the main concern is the vasculature in the heart. Now, the way you inhibit this, and all standard protocols involve some form of immunosuppression in that first phase of the trial. So even a few days before you get the virus, which is given as an IV infusion, there's a certain degree of immunosuppression because you want to minimize any of this happening, okay? But you still need to monitor for this over the first month after the treatment. So that's one type of potential side effect just from the intact capsid here. So what happens? It binds to the membrane here, and then it gets taken into the cell, into a little bubble called an endosome. And the other thing besides recognition of the cell that's really neat for this capsid is it knows how to escape from that. And that's how viruses get into your cells and infect you. They have ways of getting in. And this is the way AAV can then escape from this endosomal capsule. And then it can diffuse, it can go into the nucleus. So this is dotted line because the nucleus membrane has pores. And so you can go from the cytoplasm, which is all the part of the cell outside the nucleus. It can actually get inside the nucleus, which is where all the rest of the DNA is. And where all the action is in terms of gene expression. So it can get in there, and then there are ways that the capsid then, there's no more use for the capsid, and so it gets uncoated. It gets broken up into smaller pieces. And then what you're left with is this DNA that's inside that can now circularize and it forms like a little mini chromosome in the nucleus. And now the thinking is that this can hang around here for years. It's like you've delivered a gene, and it's just as if it's like one of your genes, but it's not actually integrated into your own DNA, and that can be an advantage in that there are other types of gene delivery where it does integrate into the DNA, and then there's always a risk it could go in somewhere where you wouldn't want it to, where it could cause a cancer or it could disrupt another gene. So this is in some ways a safer way of delivering a gene to the nucleus. So once you've got this gene here, then, like I said in the previous slide, the DNA directs transcription into an RNA. Now here, this is not the DUX4 RNA. This is our treatment RNA, and that could be different things. It could be a microRNA that then targets DUX4 RNA. So it could be an RNA that's complementary to DUX4 that then can seek that out and target that. Or it could be in terms of the Epicrispr group, it could be an RNA that can lead to the production of an entity that can then go in and trigger greater methylation at the DNA locus for DUX4, which is exactly what we want. But anyway, you get an RNA from this, and then the RNA goes out in the cytoplasm. This is the ribosome, the protein factory of the cell, and all it does is it translates this RNA sequence into a sequence of amino acids and starts making a protein that corresponds to that. All of your proteins in your body are made this way. And so in here, this is an example from a paper talking about a different disease where the product they wanted to make was called a protein called LAMP2. And so this is your transgene product, and it might be a CRISPR-Cas construct that would then target the DUX4 locus. Okay, so this looks great so far. But then the question is, there are some potential immune problems with this, in that these little pieces from the capsid, they eventually get sent to, it's sort of like a recycling machine in the cell called the proteasome. And it takes bigger pieces of protein debris and it threads them through here and chops them up into tiny little, like 7 amino acid sequences, little tiny peptides. But because there are many different types of defenses against infection. One of the types of defenses against infection is that certain bacteria, for instance, would chop up. If those are chopped up, then the protein products from those might be distinct. And there are ways to recognize, for instance, if there's some novel sequence or something that could be an invader, based on taking a subset of these little protein fragments that come out of this and actually presenting them, binding to another membrane here, going out through pathways that lead to bringing it out on the surface of the cell, where there are these immune cells called T cells that are always floating around in your bloodstream or even inside muscle. And if they recognize something novel or something thought to be an invader, then it turns on the activity of these T cells. And that's one thing we get concerned about, because this capsid is an invader in one sense.
So Larry, you have about 2 minutes.
Yeah, okay, I'm almost done. And so I just wanted to point out that the other toxicities we're concerned about are from this later T cell activation, and there are ways we can monitor for that. So, but that's where we get concerned about liver injury and potentially reaction to the transgene itself. So that was that, and I just want to go back here to this outline here. So eligibility is something we want to, in general, for each trial, we have to tailor the eligibility to what we might be concerned about. So we want to know, if someone has a significant cardiac condition on top of their FSHD, not necessarily from the FSHD, but we don't want to give a treatment that could potentially lead to worsening heart failure or things like that. If there's anemia or some other medical condition that could put the patient in danger depending on the therapeutic modality. There's things we need to test for, say, in an AAV trial. Do they have pre-existing antibodies to the AAV, as was mentioned earlier? Is their liver in good functioning state? But there are other things. If a patient's, some trials require an MRI to be done multiple times or a muscle biopsy. And if a patient's not really able to do that, then for that particular trial, they might not be the best candidate. And then there's also, you know, some degree of disability so that we would may be required for to be eligible, just so that we would have something to know that there might be something that we can follow in terms of progression. So there are a lot of questions about eligibility. Safety I already went through, and you know but the point is there's all sorts of monitoring that goes on for all of these trials just to make sure, you know, the liver's functioning well and along those lines, and that some of these complications are avoided, and that we make adjustments as needed. And then in terms of long-term follow-up as well, I mentioned about the open label extension for many of these trials. But for gene therapy trials, especially, there might be a quite longer follow-up for 5 years or so, just to make sure everything's going well. We don't know how long the transgene may be active. Hopefully, it would be for years. But the safety could potentially be an issue even for a longer period of time. And so there are sort of a lot of this information is always discussed with the patient in what's called the informed consent, where we just go through detail by detail about what the goal of the trial is, how it's going to be implemented, and what sort of things will be monitored. So people will probably have very specific questions, and I'll stop here so that we can move on.
All right, thanks, Larry. All right, Wes has slides for us as well, and he'll be talking very specifically, I imagine, about the Epicrispr therapeutic.
Yes, thank you. Let's see, are you able to see my slides?
Yes.
Super, cool. Well, thank you so much. Again, my name is Wes Miller, and Beth referred to me as the Chief Scientific Officer. I'm glad you think so highly of me, Beth. I'm actually only the Chief Medical Officer, just a doctor, over at Epicrispr Technologies. And I'm delighted to just give a very high-level overview of EPI-321, which is our investigational gene therapy one-time treatment for FSHD. So the topics covered in this hopefully very brief presentation this morning are going to be to review our understanding of FSHD as an epigenetic disorder, how EPI-321 is designed to correct that epigenetic defect, to think about some of the unique aspects of a gene therapy approach for FSHD, like as conveyed by EPI-321, a summary of the key safety and effectiveness findings from laboratory testing of EPI-321 that preceded our entry into clinical trials, and then a very brief overview of our first in-human clinical study. So it's now well accepted that FSHD is in fact an epigenetic disorder, and it's really a defect in the epigenome of patients affected by FSHD that leads to the abnormal expression of DUX4. And we, of course, know that DUX4 protein is toxic when it is made within skeletal muscle cells. So healthy skeletal muscle cells do not make DUX4 protein. Why is that? Well, the reason is shown in this figure. We know that a healthy DUX4 gene is flanked by a really large repeat array of DNA sequences called the D4Z4 region, which is depicted in this figure as these DNA units shown as triangles. What's also known on healthy DUX4 alleles is that this region is well methylated. This means that many of the DNA units within the D4Z4 have a methyl chemical tag attached to them. That's depicted as these closed blue circles. There are some that don't. They're so-called unmethylated, depicted by open, but by and large, most of them are methylated. So a general concept in human genetics is that when a gene is flanked by DNA that is well methylated, that gene tends to be silent within the cell. That means the cell does not express that gene and make the protein that it encodes for. So we now look in this figure at a person who's affected by FSHD. And of course, they've inherited a DUX4 allele and gene that's quite different. The D4Z4 flanking the DUX4 gene in this case is dramatically contracted. It's really shortened. In addition to that, we know that many of the DNA units within the residual DNA in the D4Z4 do not have a methylation tag, so they're so-called not methylated. So overall, the D4Z4 is relatively hypomethylated in the FSHD state. And so again, a general concept in human genetics is that genes which are flanked by DNA that is not well methylated tend to be expressed. That is, the protein product, in this case, DUX4 protein, tends to be made. So to review, what drives FSHD is a disease-causing mutation that leads to a contraction of the D4Z4 DNA region that flanks DUX4. This contraction is also associated with low methylation on that residual DNA strand and DNA units within the D4Z4. Genes which have low methylation are susceptible to being expressed within cells. And by the way, sometimes we refer to the status of methylation, present or absence, as the epigenetic characteristics of that gene. And so we say FSHD is an epigenetic disorder. So how is EPI-321 designed to try to address this? Well, as Larry and I think Lucienne both referred to earlier, it's designed to really attack FSHD at this epigenetic root cause of the disease. As shown in the cartoon at the bottom, there are three key components for EPI-321. The first is shown as a purple key. This is called the guide RNA. So this is a strand of RNA that's designed with a specific sequence so that it can recognize one unique area of DNA within all of our genome. And that DNA is right next to the D4Z4. The second of three components in EPI-321 is depicted as this teal-colored blob, which we call DCAS onyx. You can think of this as a protein anchor. It has a very high affinity for the purple guide RNA, as well as a high affinity for the DNA sequences that flank the purple guide RNA. And so it sits down on the DNA. Fused to it is the third of the three components of EPI-321, which is depicted as these blue spheres. So these are methylators. These are proteins that essentially work to remethylate the DNA units that are nearby where the guide RNA and the DCAS onyx anchor are sitting. So you can see at the end of the day, what EPI-321 is expected to do is to remethylate these poorly methylated DNA units within the contracted D4Z4. And if we contrast that against the very top of this figure, that is the healthy DUX4 allele, we're not trying to change the DNA sequences themselves. We're just trying to restore a healthy epigenome to be able to suppress DUX4 definitively. So EPI-321 is a form of gene therapy. And if we double click on that, what does the medicine actually look like? Larry has reviewed this very well already today. So there's two key components to this gene therapy. The first is the so-called vector, and the second is what's called the active cargo, which is the artificial engineered transgene. So starting with the top, the vector, this is depicted as this purple polyhedron figure at the very top. We have chosen to use a vector, again, called AAVrh74. This is a vector that has been well studied and well used, again, mostly by Sarepta's products, including Elevidys. And so we know that it has a predilection for being able to find and target and ingress into muscle cells, which is, of course, where FSH disease is. Another advantage of AAVrh74 is that it is not quite as endemic in humans as some of the other AAV subtypes. And in fact, we've conducted a study in symptomatic adult FSHD patients in the United States and New Zealand, tested up to 60 adults, and we have found that only 17% of these symptomatic FSHD adults have evidence of a prior exposure. So that means that we expect most patients with FSHD would be eligible to get an AAVrh74 vectorized gene therapy. At the bottom, we now see what's packaged or stuffed into this vector, this viral protein capsid, and this is our active cargo or our engineered artificial transgene. So as mentioned earlier, often when we think about gene therapies, they're really gene replacement therapies. That is, there is a disease we're trying to treat, and that disease is caused by the lack of a functioning protein. Examples would be Duchenne muscular dystrophy, where the protein is called dystrophin, or spinal muscular atrophy, where the protein is SMN. In those particular cases, the engineered transgene is just providing DNA code so that the cell can make the actual version of the missing protein or a substitute version of that missing protein. In our case, there's not a missing protein in FSHD. So this transgene is just encoding the cell to make those three exact tools that I reviewed on the prior slide. That is the guide RNA, the decasonics anchoring protein, and the fused proteins, which help to remethylate the D4Z4 region. So one of the unique aspects of EPI-321, and in fact, most AAV-delivered therapies, is that a single intravenous dose of EPI-321 gene therapy is expected to be durable. And again, this has been touched on already by both Lucienne and Larry. So why is that? And there's a couple of key reasons. The first is there's accumulating and mounting evidence that AAV vectors can persist for quite a long time within muscle cell nuclei. And as Larry showed on his cartoons, the persistence of that transgene DNA allows for that DNA cargo to be transcribed and translated. And so the therapeutic protein, in our case, our methylating machinery, is continuously expressed over time. And it's important to note that it is expressed at all times, and that might be key in a disease like FSHD. The second reason is that scientists at Epicrispr have also done studies or conducted studies with EPI-321's active component, so the decasonics anchor, as well as the fused modulators. And they have shown that even when those engineered gene is purposely removed from a cell system, the methylation marks that were deposited on the DNA when it was there remain over time. And they are even passed down through generations of cell division. So this is kind of a unique aspect of epigenetic editing that really isn't present for simple gene replacement strategies. And so thirdly, I would just sort of highlight that sometimes we think about this one-time dose nature of gene therapies as being mostly beneficial because it's convenient. But again, if we think about the way that FSHD works, that is that any DUX4 that leaks out can be toxic to muscle cells. It might be that providing this constitutive and continuous and definitive silencing of DUX4 over time might confer a real good benefit to patients with FSHD. So we're really optimistic about that. Prior to initiating our first in-human clinical trial, EPI-321 underwent extensive laboratory testing, sometimes called preclinical testing. It was the results of all of these laboratory and preclinical testing that were provided to health regulators, such as the FDA, who gave us permission to initiate our first in-human clinical studies. So I want to briefly review some of the key findings from all of that preclinical testing. First, thinking about efficacy or effectiveness, we of course measured the ability of EPI-321 to methylate. We know based on some elegant studies of FSHD families that only modest differences in methylation can mean the difference between clinically severe disease and completely asymptomatic patients. And so we were targeting a roughly 5% increase. We were able to see that we consistently met this across many FSHD patient-sourced cell lines that were exposed to EPI-321. Of course, and logically, we also measured the ability of EPI-321 to suppress DUX4 as well as the DUX4 pathway. Some of the best animal models of FSHD have given us a sense that suppressions greater than 50% might have significant impact in real human disease, and we were able to see this deep suppression across a large number of FSHD patient-sourced cell lines. Apoptosis is the final pathology of FSHD. That's a fancy medical term for saying the muscle cell dies. And so we know that EPI-321 is able to reduce the abnormal apoptosis rates in FSHD patients when we treat them in the laboratory. And finally, we did lab studies where we created three-dimensional engineered muscle organoids that were sourced from FSHD patients and studied the contractile force that they can generate. Of course, we know that in these untreated FSHD patient-sourced organoids, they are weak compared to healthy muscle. However, when we treated them with EPI-321, we were able to see that we were able to increase the contractile force to normal healthy muscle organoid levels. Switching to safety, I want to highlight some of the key aspects of our safety studies. First, there's a hypothetical risk when using this approach that there could be so-called off-target gene suppression. That is, Could Epi321's machinery bind another gene besides DUX4 and inadvertently cause suppression of that other gene? We wouldn't want that to happen. And so we did extensive testing preclinically on human muscle cells, human non-muscle cells, mouse muscle and non-muscle cells, as well as monkey muscle and heart muscle cells, and saw no evidence that Epi321 binds to and suppresses any other gene except for DUX4 and within muscle. Second, there's a possibility that the anchor protein that's used in Epi321, which is called DCAS-Onyx, could elicit an immune response that could be injurious to muscle tissue. And so we tested mice and monkeys and followed them for up to 6 months after very high IV doses of Epi321, and were able to see no evidence of this. And finally, we conducted overall safety studies, so-called toxicology studies of Epi321. Again, these were conducted in mice and monkeys. They were followed for up to 6 months after high intravenous levels of dosing. And this was observed to be safe in monkeys and mice, including at doses up to 6 times our planned initial clinical dose. And so finally, I want to transition briefly to a high-level summary of our ongoing first-in-human clinical trial. We are expecting to enroll and dose up to 12 adult participants with genetically confirmed FSHD type 1. That genetic confirmation will occur on the study. It is multi-center and multinational, with trial sites expected to participate from the United States, New Zealand, as well as Australia. Adults aged 18 to 75 years of age at the time of dosing will be allowed. As mentioned prior, we have to test these patients to make sure that they are AAV-RH74 immune negative. That is, that they don't have memory. If they are positive, they would not be eligible to get the investigational gene therapy. These patients have to be able to take and commit to taking immune suppression for several months following dosing. As mentioned prior, we do extensive and very focused and detailed testing on vital organ function. So this is heart, lungs, liver, and kidney of all participants prior to them being enrolled. As mentioned, this is a one-time dose. However, it's important to note that the FDA and other health regulators for all gene therapies require a so-called staggering approach. This means if we're aiming to enroll and dose 12 total participants on our trial, we can't do them all at the same time. We have to dose patients, wait some amount of time, dose the next patient, and so on and so forth. And this is, as Larry said, we can't take the medicine back once it's given. And so we're trying to make sure there aren't any unexpected side effects that might arise. If they do, that gives us a chance to stop, figure out how we need to course correct before we dose or expose other participants. So interestingly, on our study, we're planning 2 clinical doses. They're listed here. And it's perhaps noteworthy that these are significantly lower than most other AAV-based doses that are used for neuromuscular diseases. So for instance, this initial starting dose is less than 1/6 of the dose of Elevidys. That is the other approved AAV-RH74 product that's in the clinic for Duchenne muscular dystrophy. And there's a reason that we think and anticipate that lower doses are going to be beneficial for FSHD. And that's because, as I believe Lucienne was referring to earlier, we're not having to provide large amounts of transgene copies to be able to continuously make massive amounts of a deficient protein. Instead, we simply need to assemble sufficient machinery to be able to bind that one abnormal gene, methylate it one time, and then to be done with it. Finally, there's approximately 5-year follow-up, and it's for the reasons that Larry stated. So I'll stop there because I know we're probably running short on time, and I'll turn it back over to Lucienne.
Thank you, Wes. That was really helpful. And we do have questions that have come up while you and Larry were talking. And you addressed one of the questions by describing that 2 × 10⁻³ and 4 × 10⁻³, I think, viral grams per kilogram of person weight is how you will decide to dose. And that was one of the questions, was how will you decide what dose to give each patient? And it's really based upon your weight, the patient's weight. And as Wes was describing, I believe you derived the dose from your preclinical studies. It's 1/6 of what Sarepta gives with Elevidys, but it's also derived from your preclinical work.
Right.
And so these doses that you're saying that you're using currently, are these the doses that you used in your preclinical work that were shown to be efficacious? We've had that question.
Yeah, so the short answer is yes. So VG per kilo is vector genomes per kilogram. And so there's now over 80 humans who have received AAV-RH74 by way of Sarepta's products, for whom data has been published. When they take the muscle biopsies, we know exactly how much vector gets in. We were able to triangulate that information against our preclinical program, where we were able to measure how much did we need in these systems to be able to deeply suppress DUX4, rescue apoptosis, rescue contractility. And then we basically just triangulate and make the leap to say, okay, these are the clinical doses we need to use. That's exactly it.
Thank you. That's great. And how will you measure whether or not you've methylated that region enough in a patient who's received the therapy? How will you measure that?
Yeah, so sometimes I think if I were an FSHD patient participating in a clinical trial, I'd be like, why are they asking me to do all of these things? And one of the things we ask them to do, of course, is to provide muscle biopsies. And we're not just doing that because that's fancy science. We're doing that because that's really the way to understand if kind of at the root intended mechanism of EP321, is it doing what we think it's supposed to do? And so when you collect muscle biopsies from 12 patients, you start to get a tableau of how is that working? What's the average increase in methylation? Is there a range? Does it matter if they got 2 versus 4 times 10 to the 13th, right? All of these things sort of help us to sort that out before we move on to kind of the next phase of clinical development.
And how quickly will you know there's effect? There's one question that said, well, will you have to wait during the entire 5-year evaluation period to know if you have an effect? But how quickly do you anticipate you can ask that question?
Yeah, so I think I'd certainly, sorry, Larry, I'd love your opinion on this too, but I think the question about how can you know if a treatment is helping patients depends on the underlying disease you're treating, as well as the nature of the treatment that you're giving. And I would say for FSHD, we're probably looking in the 6 to 12 months or potentially even longer time range. But I think 12 months is probably a good educated guess right now. To be clear, all gene therapies require at least 5-year follow-up. Some of them require by the FDA 15-year follow-ups. An example would be Casgevy. It does require 15-year follow-up. This is the only approved gene therapy for sickle cell disease. That doesn't mean that it took 15 years of all patients on the clinical trials before they were able to submit. They actually submitted for approval much more rapidly than that. It's just they have to be followed to make sure there's not later onset safety signals that evolve.
Thanks,
So
Wes.
for EP321, we would expect to be able to submit prior to the very last patient getting through 5 years. Yeah.
You exactly are addressing the questions very clearly, and that's awesome. I think people really appreciate that. Some folks are wondering about being able to see the efficacy and safety data in your animal models. Is that already published, or is that available somewhere for people to review?
That's a great question. So we have. And we've presented quite a few at quite a lot of meetings, and so there's different data that has been presented in different places. I don't believe we have a holistic singular paper that would. I mean, I can tell you it was like 2,000 pages that were submitted to the FDA. So, but certainly I can get back to the society to see if maybe there's a publication that we have submitted that I'm not aware of. So I can follow up with you all about that.
That's great. And I think I found some publications on their investor site, and that we could perhaps provide links to that through the society in the future. Let's see. Okay, so we have, in which countries are the studies ongoing? We had a person living in France asking, are they able to participate? And so could you tell people where the study is happening?
Yeah, so for our study of EP321, the United States, New Zealand, and then Australia are expected 3 countries for this first in human N of 12 study. But there are other studies of other investigational products for FSHD that are underway, including some in Europe. And so I would encourage people, if they live in France, to certainly look into that. Clinicaltrials.gov is a really good resource for finding out that information.
And people are interested about pre-existing conditions that might disqualify them from a study. Someone mentioned hemochromatosis, and perhaps that's not a disqualifier, but what are the disqualifiers?
Yeah, so that's an interesting one. It's impossible to list out every known medical disease and condition, but I would say hemochromatosis would be one that we would have to give really careful thought to, because there can be liver injury that's involved in that disease, depending upon how advanced it is. or how well treated it is or isn't. So I wouldn't say for sure that having hemochromatosis makes you ineligible, but we would have to really think long and hard about that at a per patient level.
Okay, and two more, I think, really important questions, and then we're going to run out of time. How long would one be on a steroid? And could muscle weakness occur to being exposed to a steroid?
Yeah, that's very good questions. So it's really interesting. If you take people who do not have muscle diseases, myopathies, muscular dystrophies, and you give them long course steroids for other reasons. So my background is I'm a pediatric oncologist, so I've treated lots of children with leukemia with long courses of steroids to try to treat their cancer. And, some fraction of those kids will actually show a steroid-induced myopathy, we call it, which is a muscle weakness. Conversely, there are some muscular dystrophies where steroids seem to be beneficial. This is obviously most exemplary in Duchenne muscular dystrophy. I don't know that we have good evidence one way or another for FSHD. There's one publication that seems to suggest, it was done by Robbie Twill's group back in the 90s, I believe, that seemed to suggest steroids don't seem to help. But it also didn't seem that they hurt. So, I think probably, and Larry, let me know if you disagree, but it's probably neutral as best that we know in FSHD.
Yeah, I would agree completely. I mean, we sometimes see patients in the neuromuscular clinic who are on steroids and they get some proximal weakness. And yet if they're able to stop the steroids, then everything usually reverses. And what generally is talked about in this type of trial is a early dosing with steroids for usually less than 3 months, almost certainly less than 6 months, just to make sure that those acute toxicities that I talked about aren't occurring.
Yeah, so Lucienne, for the duration question, we pretty much copy what's done for Zolgensma and Elevidys. So it's 2 months at full dose of steroids, and then it's a week-by-week taper, usually over the course of another 2.5 months.
Great. And we'll end on this question. How would a patient apply to be included in the clinical trial for EPI 321?
Yeah, so the best way to do it is to go to clinicaltrials.gov to look for the various sites that are posted as active in recruiting to find one that makes sense for you and to contact the study coordinator that's listed.
All right. So those were great questions. Thank you, everyone, for sending them. Beth, I'll turn it back over to you to just close this up.
Awesome. Whew, that was a lot of information. I don't know about you guys. My brain is full now. Thank you. So thank you, Doctors Ronco, Hayward, Miller, for your time and presentations today, and for honestly taking the time to answer all of our questions. Our community is so grateful and lucky to have you in our corner. So thank you guys so very much. Our next and final FSHD University webinar for 2025 is going to take place next Thursday on December the 18th. This is a good one too, guys. Join us as we take a really deep dive into the latest progress in FSHD drug development, including Epicrisprs. It's going to be presented by Dr. Lawrence Korngut. He's an associate professor of neurology and clinical neurosciences at the University of Calgary in Alberta, Canada. We also encourage you to visit our website events calendar for all of the upcoming events, both in your area and online. Thank you once again for joining us today, and we'll see you next time. Thank you, everyone. Bye-bye.
Okay. Bye, everyone.